Assalamualaikum and a very good day I bid to Dr. Fairuz, Dr. Faiza and to all the other viewers of this video. In this video, the contents of the research proposal will be discussed in order to instill understanding on the proposed research, which is a modification of alginate microcapsule to suit the natural environment of islets in type 1 diabetes mellitus patients. Type 1 diabetes mellitus, or known as T1DM, is an autoimmune disease that is caused by the destruction of pancreatic beta cells, causing the hormone insulin production to be decreased. T1DM patients often suffer from hyperglycemia, a condition in which the blood's insulin level is too low, causing glucose level to elevate, as insufficient insulin available to level the blood glucose to an optimal level. To combat this problem, T1DM patients opt for daily insulin intake to maintain their blood sugar at a normal level. However, exogenous insulin injection also comes with a consequence, known as iatrogenic hypoglycemia, a condition of abnormally low sugar in the blood. This often leads to hypoglycemia-associated autonomic failure. The previously presented solution to this problem is islet cell transplantation through microencapsulation delivery method at the target area. The purpose of encapsulating islet cells into a microcapsule is to protect the islets from the action of immunosuppression of the patient's immune system onto the donor islets, which helps reduce the dependence on immunosuppressive drugs. The microcapsules for drug delivery systems are usually made from biocompatible materials such as polysaccharides that can form hydrogels. Most commonly utilized polysaccharide as capsules is alginate due to its low toxicity, low cost, and gelation capacity through the addition of divalent cation, calcium, and barium. Alginate matrices possess a special characteristic in which they are structurally identical to the extracellular matrices of living tissue, making it favorable for cell transplantation in a less invasive operation. Alginate hydrogel for microcapsule can be prepared through a cross-linking method of divalent ions with aqueous alginate solution and the outer pore size of the alginate hydrogel minimized by coating the alginate microbeads with polyamino acid. This research is proposed because of the presence of multiple gaps in clinical trials in previous literature. According to Colmer and friends in 2016, alginate has been the most widely accepted encapsulating material due to its ability to provide capsule stability, biocompatibility, and reproducibility. However, the results from clinical trials were never promising. A study conducted by Calafiore and friends in 2006 resulted in the failure to achieve lifetime therapy for type 1 diabetes patients. Another study by Tush and friends in 2009 demonstrated that if allotransplantation of encapsulated human islets procedure was not significantly improved, it might cause an inflammatory response or ischemic necrosis in the patients who did not rely on immunotherapeutics but on the anti-inflammatory drugs only. In 2016, Colmer and friends deduced that the failures in clinical trials were mainly because of deficiency in reproducibility and incompetency in reaching insulin independence for a longer duration. Failure in islet grafting could be due to the undesired reactions from the encapsulating materials. Therefore, a study on the improvement of biocompatibility of the alginate-based matrix to the islet cell grafting for T1DM needs to be conducted. Hence, this research is proposed with the objectives to produce alginate microcapsules that can imitate the natural environment of islet cells, to examine the biocompatibility of modified encapsulated islets with extracellular matrix molecules, and to estimate insulin production from the modified encapsulated islet microcapsules. The proposed solution involves modification of the alginate-based microcapsule to suit the natural environment of islets in type 1 diabetes mellitus patients. Firstly is the decellularization process in the preparation of the extracellular matrix, in which a human pancreatic tissue will be withdrawn from an adult diabetic donor and placed inside an ultra-pure endotoxin-free sterile deionized water, followed by the addition of deoxyribonuclease 1 from bovine pancreas in magnesium chloride solution. Then, the mixture will be shaken, then further projected to incubation in eta-trisma-based solution for 18 hours on a shaker with 200 revolutions per minute at 4 degrees Celsius. Sterile deionized water will be used to wash the tissue, and the tissue will be shaken under the same parameters. Lastly, in order to acquire a fine ECM powder, the resultant wet scaffold will be frozen at -80 degrees Celsius, undergo lyophilization and cryoablation. Next step in matrix preparation is solubilization of pancreatic ECM, which will follow the reported procedures from previous study with several modifications. The lyophilized ECM powder will be solubilized in hydrochloric acid with pepsin and stirred constantly for 48 hours at room temperature. The pepsin is inactivated permanently by maintaining a pH 7.4. The solubilized ECM will undergo centrifugation and the collection, freezing at negative 80 degrees Celsius, and lyophilization will be done with the supernatant. After the lyophilization, the supernatant will be converted into a fine powder, then vialed in glass ampules, sealed and treated under gamma radiation. The alginate preparation is initiated by mixing and stirring the controlled alginate solution with Henk's balanced salt solution. For the ECM alginate solution preparation, the solubilized ECM will be diluted with the HBSS. Rheological measurement is needed to measure the mechanical stiffness of the alginate matrix. The steps for alginate discs and non-decellularized human pancreatic tissue preparation for rheological analysis will be performed as described in previous literature with a slight modification. First, the alginate discs will be prepared by placing high manuronic acid (LVM alginate) in a 48-well plate, followed by addition of the cross-linking solution strontium chloride. Strontium chloride is a better alternative to the commonly used calcium chloride, as the calcium ion crosslinker is reported to promote the inflammatory response. The alginate discs will be allowed to crosslink for 15 minutes. The steps will be preceded with the removal of the disc from the plate wall and dabbing them gently with a Kim wipe to eliminate excess liquid. The cross-linked strontium chloride alginate hydrogel will then be placed under the rheometer for compression and storage modulus determination. The next step in the modification is the islet encapsulation and culture, by which the previously isolated islets will be mixed gently with the alginate solution. The microbeads will be obtained by the external gelation method. The microcapsules will be collected in strontium chloride with HEPs and will be allowed to cross-link in two conditions, with the incorporation of ECM and without NCM. Pseudo-islets will be formed within the microcapsule by incubating the microcapsule containing pancreatic single cells with sodium citrate solution. The mixture will be agitated for 5 minutes to obtain a liquid course. The dimension and morphology of alginate microbeads will be evaluated by an optical stereo microscope. Microcapsule size and distribution will be determined by photomicrograph analyses. The mechanical stability of the microbeads will be assessed by the induction of mechanical stress using bead agitation by an orbital shaker. The microbeads will then be examined under an inverted microscope to assess any beads damage and count intact beads. The mechanical stability can be translated into the capability of the ECM incorporated islet encapsulation to resemble native ECM and withstand biological cues for islet survival in our body. The quantification of early apoptosis will be assessed using apoptosis detection kit as described in Hensley and Friends in 2016. The encapsulated islet will then be quantified using Beckman-Coulter BD Fax Canto 2 flow cytometers set at 488 nanometer. The data will be analyzed using FlowJo LLC software. The quantification of cell viability will be conducted following a protocol described by Cañabano Hernandez and Friends in 2019. Cell viability of encapsulated islet will be quantified using live-dead viability cytotoxicity kit. The encapsulated islet will be released from the microcapsule and quantified after incubation with Beckman-Coulter BD Fax Canto 2 flow cytometer. Data will be analyzed with FlowJo LLC software. The cell viability will be monitored by microscopy imaging under a Nikon TMS fluorescence microscope at 495 nanometer. Random images will be analyzed with the EclipseNet software. Membrane integrity of encapsulated islet will be determined by the in vitro toxicology assay kit lactic dehydrogenase. The supernatant will be incubated with a kit cocktail mixture in absence of light. The color development will be quantified on an Infinite M100 Tikon microplate reader at 490 nanometer with a reference wavelength of 690 nanometer. The impact of the modified ECM on islet functionality will be tested using the glucose-stimulated insulin secretion test. The insulin content will be quantified by Perkin-Elmer insulin alphalysa. The glucose stimulation index will be calculated from the ratio of insulin release during the high glucose phase to the insulin release during the low glucose phase. The responsiveness of the islets can be portrayed by the glucose concentration. The results presented in this study will be expressed as means plus minus structural equation modeling SEM of 3 replicates. Statistical comparisons will be performed using IBM SPSS Statistics 18.0. One-way analysis of variance, known as ANOVA, will be used for analysis, followed by Tukey's HSD post hoc test in case the values presented significant differences with statistical significance set at a level of 0.05. In conclusion, the modification of alginate microcapsule to suit the natural environment of islets will be performed to provide better treatment for diabetes type 1. The study will deliver a number of benefits to the diabetes type 1 patients, scientific research society, and to the overall public. Through the transmutation of modified encapsulated islet cells, diabetes type 1 patient's body will be able to produce its own insulin once again. The damaged beta cells of the pancreas will be replaced by the modified encapsulated islet cells, which will help the patient in normalizing the blood sugar level via insulin production, and daily insulin injections will no longer be needed. This study will also help the Scientific Research Society by providing an enhanced method of the preparation process of biocompatible modified encapsulated islet cells, which could be further utilized in future studies of drug delivery and stability. Furthermore, this study will help to highlight the importance of using enhanced encapsulation technologies in drug delivery to discover more convenient treatments for several chronic diseases, including cancer, atherosclerosis, myocardial ischemia. This study is for the betterment of humankind and will help to prevent the loss of human lives to such chronic disease by providing more effective and efficient treatments. Thank you for allocating your time in watching this video and listening to this narration.